Welcome back, everybody. Our next speaker is Cuomo from the EBI, and she will talk about genetic analysis of differentiating cells using single-cell RNA-seq.
Okay, thank you. So today I will tell you about an ongoing project where we combine, is this working? Yes. Single-cell expression profiles and human genetic variation to explore the effect of common genetic variants on transcriptional variability. So just to give a little bit of background and motivation for this project, so a major challenge in biology is to unravel the genotype-phenotype map and understand how DNA variants might shape observable traits such as height or eye color and cause complex diseases such as diabetes, IBS, cancer even. So this is exactly what GWAS do. So genome-wide association studies aim to find these types of associations between genotype and phenotype. So in a classic GWAS, samples that all share one phenotypic trait, say diabetes, are pulled together alongside a cohort of healthy individuals that will act as controls, and then one looks for associations between SNPs or common variants and having the trait. And they've been widely successful at that. I think those are numbers from 2017, but there's been more than 15,000 different associations being found with almost 1,000 between traits and diseases. However, the interpretability of GWAS hits is often far from trivial, especially when those hits are sitting on non-coding regions of the genome. And so one way of trying to understand the molecular mechanisms that link these two parties, those the genotypes and the phenotypes, is to look at intermediate molecular phenotypes, such as gene expression. One possible mechanism is, for example, that having a certain letter at a locus, a G here, might increase the affinity for a transcription factor to bind and thus boost expression. So, all the individuals that will have a G at that locus will have, on average, high expression of this specific gene. And methods that find associations between genotype and gene expression are called Qtls, or expression quantitative trait loci. So those loci that affect one quantitative trait, in this case expression. Right, so traditionally Qtls used as a measure of gene expression bulk RNA-seq, which means that all cells from one individual are pulled together, all the mRNAs extracted from those cells and mapped onto a reference genome. So this results into one individual value, expression value, per sample, per gene, that is essentially averaged out across all cells. And the way we normally visualize an Qtl is depicted there in the bottom right corner. So we'll look at the expression of a gene of interest. And this is bulk, so every dot here is a donor. And then we stratify our donors by their genotype at the SNP we're testing and look for a slope along the means. So in this case, the more G alleles, the higher the expression. And so, it's important to note that in order to maintain statistical powers, since we're often testing thousands of genes, we do not scan the entire genome for SNPs, but only look within a window around the gene. This is also because we do expect most regulation to happen within the vicinity of the gene. So we refer to these as cis Qtls as opposed to trans Qtls that would have an effect on a long, larger distance. We also often only look at common SNPs. So SNPs are present in at least 5% of our population. Finally, to map Qtls, we fit a linear mixed model that, in practice, the linear side of the model essentially finds that slope that I was describing before, and whilst accounting for the population structure of our samples. Recent advances in experimental techniques, as you all know, allow us to assay those same molecular traits at the single cell level, potentially opening new avenues. However, we also do know that this data is a lot noisier. So single-cell RNA-seq data is a lot noisier than bulk. And so the very first thing we need to check is, can we even map Qtls? Can we even do this? Or is this so noisy we could never assess differences in expression? So for example, can we reproduce known Qtls that were previously found in bulk? And then assuming we can we do more? Can we leverage this increased resolution to define new phenotypes entirely? Okay, so let me show you, let me introduce the data we use to look into this. So this data was generated by the Ludovic Vallières lab at the Sanger Institute, just next door from us in Cambridge, UK. And it was generating using HIPSCI lines. So HIPSCI is the Human-Induced Pluripotent Stem Cells Initiative, which is a very large project that aims to characterize those type of cells. So those are human iPS cells. Those are originally fibroblasts, skin cells. They were then reprogrammed into becoming stem cells or behaving like stem cells. And in particular, we used samples that were then from one of the differentiation studies that followed from Hipsky. So our cells are actually differentiating from iPS into definitive endoderm. Endoderm is one of the 3 germ layers alongside mesoderm and ectoderm. And endoderm gives rise to most of the internal organs, such as the liver, the lungs, the pancreas, the guts. Right, so it's a rather short protocol. It's only 4 days. So we collect data at day 0 first, so when they're still iPS cells, and then every 24 hours after triggering differentiation, so day 1, day 2, day 3. We expect them by day 3 to have reached the definitive endoderm stage, whereas at around day 1, they should be in this bipotent state where they've given up the ectoderm fate, but they're still bipotent for both mesoderm and endoderm. And within the Hipsky resource, we have imputed genotypes, so SNP arrays for all of those individuals. One important thing to note is that part of the experimental design, cells from 4 to 6 different donors are actually grown together in a max population. And this allows us to avoid confounding between technical batch effects and genetic donor effects. And because we have the genotype, we can always trace back what donor the cells were coming from. Okay, so just to break down the main sources of variation within this data set. First of all, of course, there's population variation. We're looking at SNPs. These are humans. Second of all, there's this huge process that the cells are undergoing. They're going from being pluripotent to committing to one specific fate. And so all sorts of molecular changes are happening. And finally, thanks to the single cell resolution, we have access to cell-to-cell changes in transcription. So even within one individual, one stage of differentiation, we still will have differences in terms of transcription from cell to cell. In terms of numbers, we have about 40,000 cells, human cells from 130 different donors, collected at those 4 time points from iPS to definitive endoderm. Right, so first look at the data. This is a Pca plot, so it's a dimensionality reduction technique used to visualize, for example, data. And what you see here, every dot is a cell, and they're colored by the day of collection. So dark blue are cells collected at day 0, light blue at day 1, yellow and red at day 2 and day 3, respectively. And the very first thing that is quite obvious from this plot is that the first principal component, so the x-axis here, explains most of the variation in this data, and it's very well aligned with the differentiation, with this linear differentiation trajectory. And so we're going to just use this first PC as a sort of a pseudo-time. So we're going to order our cells based on this first principal component. We're going to say that low values of this will indicate pluripotent cells, and high value will indicate differentiated cells. Okay. So we can start having a look at genes that are differentially expressed along pseudotime. So there's a heat map. Every row here is a gene. Every column is a cell. Red means high expression. Blue means low. It's normalized, so that's why it can go negative. And you can see that we find 3 main clusters of cells. So if we look at the first one, the genes that are highly upregulated in these clusters are genes such as NANOX. So this is a known pluripotency marker. So as you'd expected, overall the expression is going down as time passes. And if you look at the cells that are in this cluster, so the first bar there, those are all dark blue, right? So those are all day 0 cells. Similarly, if we take the second clusters, genes like T are upregulated. This is a known master regulator for the mesendoderm stage. And again, those are mostly day 1 cells. In contrast, if we look at the last cluster, so GATA6-like genes, so those are definitive endoderm markers, we see that actually day 2 and day 3 cells are very much mixed together. So in this case, having the single cell gives us the power to look at just the transcriptome of these cells and really define, and that works as a better readout of the differentiation stage the cells are in, rather than just the day of collection. So we're going to, instead of use the day of collection as the main biological stage, we're going to use these three main stages that we're going to call IPS, Mesendo, and Defender. All right, so back to the first question. Now we want to do genetics. And what we said is the very first thing we need to check is, can we even do this, right? Can we find EQTLs using single cells? And within the HIPSCI resource, we actually have access to bulk RNA-seq for those same exact samples. So we're perfectly set to make the comparison. So we can run EQTLs using bulk expression, or we can use single cell expression. In this case, we basically take the average expression for all cells from one individual. And this is what we find. So bear in mind, these results are only on the top 4,000 something highly variable genes. The results will be on a different set of genes later on, but what's important here are the proportions, really. So first of all, we run this. So this is the linear mix model I was showing before, and now the phenotype, the expression phenotype is the mean of single cell expression. And what we see is we find about 500 E genes, so this means genes that have at least one EQTL at that significance threshold, so 10% FDR after correcting for multiple testing correction. And then we can look up those 500 in the bulk set and see how many of those replicate. And we see there's a decent replication, so about 80% of the things we find we would have found using bulk as well. So that's reassuring, right? We're not finding just false positives, we find something that we would have found with bulk as well. So if we didn't have a reference, because in this case we're using bulk as the reference, as the gold standard. If we didn't have it, still we could be quite happy with what we find and believe, trust the results. But since we do have the reference, we can also do the other way around. And the picture doesn't actually look all that good. So this is the other way around, right? So we run bulk, we find a lot more. So we find more than twice what we're finding with single cells. And on top of that, we can only replicate about 50%. So that doesn't look very good. And again, this is at 10% FDR. So that's a quite lenient significance threshold. However, if we're stricter, so let's now look at 1% FDR. So let's look at only stronger EQTLs. We see that obviously in absolute numbers, this goes down, but our replication proportion increases. And increases even further the more stringent we are. So the take-home message from here is basically the very strong signals we're always finding, even with single cells. What we do like is the power to find the weaker ones. Right. And we think we can explain at least partly this lack of power due to just lower sequencing depth, something we all know, right? Sequencing depth is lower in single cell than in bulk. So what I did here is a small simulation. So I basically downsampled the number of reads from bulk. So it starts from about 45 million reads on average per individual in bulk, and that's 100% up there. And you run it, and you find about 1,100 EQTLs, which is consistent with what's on the left. And then you take 90% of those, 80%, and so on, and you run it every time and see how many EQTLs you find. And you see that the number of EQTLs decreases quite significantly. Now, if you look at single cells, if we were to aggregate all reads from all the cells from an individual, you see that on average, you're a lot lower, right? You're about 30 million reads on average per individual, which is about 2/3 of what was in bulk. And if you look at how many EQTLs that corresponds to, or E genes again, it should be, that's a lot more comparable already to what we were finding. And keep in mind that we're not modeling the noise, we're not modeling all the other factors that change between single cell and bulk. So we're quite happy with this. What we find, we can, with some caveats, find QTLs using single cells, so we can move forward. So remember those three clusters. We had defined those three stages. So we've ran EQTLs, identified EQTLs at IPS. Can we run it? Let's identify them in the other stages. And EQTLs are known to be cell type specific. So there's numerous examples of EQTL or SNPs that affect the expression of a gene in one cell type, but not another. However, in our case, those cell types are actually quite close to each other, right? This is rather small time frame. So we want to see, can we find those differences even in those three stages that we've defined? Can we find something like that cartoon where there's no difference between A and T in the expression in the early stages, but there is in the late stage? So first of all, how many do we find at all? So as I said, there's a different set of genes. Those are all 11,000 genes that we can quantify. And this is how many QTLs we find at each of the stages. So roughly between 10 and 20% of genes that we test have at least one EQTL. This is a little bit low. In bulk, normally we are more around 30%, but still pretty good. One observation is that if we had done it using the day of collection, so day 1 and day 3 respectively, even though for similar comparable number of cells, we would have found a lot less. So we would have had a lot less power, and that's, we think, mostly because those populations are a lot more heterogeneous than the ones we can define using the transcriptomes. Oh, ooh.
It doesn't seem like there is any.
Okay. Okay. So let me show you one example of eQTL. So what I'm showing here is a different way. These are Manhattan plots. So what those are basically for one gene, that gene up there, CNT and AP2. Those are all the SNPs we're testing. So they're within a window around the gene somewhere. All those dots, every dot is a SNP that we're testing. On the x-axis is their position, in this case on chromosome 7, and on the y is their significance in terms of -log 10 the p-value. So every time we test one SNP, one gene, we get a p-value out of it. The smaller the p-value, the higher the significance. The higher will be this dot, that dot. And that yellow line is the significance threshold after multiple testing corrections. So what you see is that for this gene in the 2 early stages, there's absolutely no SNP that reaches the significance threshold. However, in the definitive endodome stage, so those 3 Manhattan plots I'm testing the same thing in each of the stages, and they're colored accordingly. There's this very clear peak in this definitive endodome. And again, we can also have a look at them in forms of box plots, as I was showing before. And the slope is flat, is near 0 in the first 2, and there's a clear negative trend in the definitive endodome. So we will call those stage-specific eQTLs. In this case, it's a definitive endodome. And this is not at all an isolated case. It's about a third in each of the stages of the eQTLs that we find are stage-specific. So this means it's significant in that stage, not significant in the other 2. Right. One other interesting phenomenon that we can identify using this approach and this data is that we can find things like this. So we call this elite switching. So what this is that we find 2 different SNPs at 2 different locations, those 2 on that chromosome 22. They're both affecting the expression of the same gene, but at 2 different stages. So this, can potentially shed light into how the regulation happened. It is well known from the literature that enhancers, for example, can happen at very specific stages. And so these types of analysis can provide a new layer to our understanding of regulatory elements and mechanisms along development. Okay. So in a way, the definition of those 3 stages, the way we did it was basically, we order our cells based on the pseudotime and then kind of binned it in 3 big bins that, correspond to key biological stages, but in practice, they're 3 bins. So something like this. For every gene, every SNP, we'll look at the QTL in IPS, IMIS in Defender. So one could try and go one step further and say, okay, what if it's more fine-grained than that? Is it maybe not just those 3 categories, but is it even like smaller time frames? And so one can start binning, the pseudotime. In this case, these are 5 bins. It's just a cartoon. The caveat here is that we do want the bins to be big enough that we have cells from enough individuals, we have enough copies of each of the genotypes to perform our genetic study. So, one solution to that is we use a running average or a sliding window type approach. So every window is still large enough, but then we slide it back a little bit. So we do end up with enough bins to try and make sense of this. Right, so what we do is basically something like this. So we have this sliding window, and then for each bin, we stratify our cells based on their genotype, and then perform QTLs, exactly like we were doing before. And so record some sort of genetic effect value, in this case, say a p-value, for example. And so you'll then have a curve, something like this, where you have -log 10 the p-value at each of the bins, which in a way represents a curve, dynamic change of the genetic effect a long time. And one may wonder, okay, what if this is just basically a response to just a change in expression, right? So you don't see the QTL anymore just because the expression is so low, you can't detect any changes anymore. So alongside this, for comparison, we also just, in that same bin, just record a measure of just the overall expression. So we don't stratify by genotypes anymore, we just take one value of expression. And you know, that will correspond, that will also create a curve, the same length. For every bin, we have a value of average expression. We have a value of genetic effect. And we want to see if they're the same, if they're different, what's happened. So these are some of the results. So let's look at expression first. So we can cluster. In terms of expression, the genes mostly cluster in these 4 clusters. So the first cluster, for example, is genes, again, red is high expression, blue is low expression. They're going down in terms of expression along pseudotime. And now let's look at the genetic effects. So those also cluster in 4 clusters, but they absolutely do not correspond to the others. So it's not like when the expression goes down, there's also no more genetic effect. Let's look at the first cluster, for example. You see that all, for one cluster in expression, still all 4 clusters of the genetic effect appear. And just to give an example, if we take one EQTL there, we see that overall the expression is going down. So what you see here, the dotted line are the 2 alleles, and the blue is the overall expression. So you see overall the expression is going down because it's coming there from the first cluster. However, most of the genetic effects and most of the largest gap from from allele to allele happens towards the end. There's also examples of exactly the other way around, right? So the expression is overall going up, but most of the change in terms of genetic happens at the beginning of pseudotime. And even perhaps more interesting or more complicated are things like this, where you know, both alleles start from the same place and in the same place. Overall, they're going up in expression. However, they're getting there using 2 very different paths. In this case, for example, the red allele is getting there a lot faster than the green. So, you know, the expression is overall going up, but most of the genetics happening in the middle of pseudotime. Yes.
So, in these static situations,
yeah,
it seems like the standard math for the significance carries through, right? So you can just use it. But what about these dynamic situations? Can you say anything about the significance of a pattern of change?
The significance in terms of p-value or?
Yeah,
so those are all significant EQTLs. We're selecting, those are only the significant EQTLs.
Right, but if you look at the actual pattern of change across pseudotime.
So it belongs to that cluster there where most of the changes happening in the middle, right? So in that case, that's minus log 10 the p-value, so red means high, so that's a big genetic change. And blue is no. How big, you mean?
Right, but isn't that change between the sort of static, the 3 stages you had before, or is it?
Yeah, so that's right. The 3 main classes, so it's 4 classes, but really it's 3 main dynamics that happen that indeed cannot correspond to going up and down, and going down, which are the 3. Right. Yeah.
And you're testing in every window?
Yeah.
And the windows are overlapping and so on?
Yeah, exactly. So that's the caveat, right? You need windows that are big enough that you have the power to detect genetics. So we find some more interesting dynamics that are not just those 3, but it's complicated.
Yeah, but you're correct for the multiple testing only within each window, I guess, or across all windows?
Across all windows.
Across all windows? Okay.
Wow. Yeah. So, yeah, you're correct first at the gene level, we use permutations normally, and then you're correct. This is about, I think, 30 windows, 30 bins.
30 windows? Oh, okay.
Yeah, it's not too many, yeah, okay. So yes, that's it.
Sorry,
yeah.
Samples in each window?
How many cells or how many
samples.
individuals?
Yeah, cell samples.
Cells, roughly, maybe, I think 2,000 to 5,000 each. Quite a lot. quite a lot of cells. And in terms of individuals, that might vary slightly. So we keep fixed the number of cells. That doesn't necessarily maintain the number of individuals, but roughly it works pretty well, yes. Yes. So yeah, just to conclude, first of all, we can perform large-scale single-cell genetics, which is, not really a big thing yet. Mainly replicating IPS Qtls, so for those we have the bulk, so we can compare. We characterize the Qtls at 3 successive stages of early differentiation, and we can identify variants that are specifically active at one stage or another. And finally, by relating complex dynamic changes in terms of genetic effect and expression along differentiation, we can start to try to understand and identify different regulatory mechanisms and processes. Right, so with that, I'd like to thank my 2 PhD supervisors and the whole groups, so Oliver Stegler and John Mariani at the EBI, in particular Daniel and Davis in all this group, the various lab that generated the data for us, in particular Maria and Nika, and the Hebsky Consortium for providing us with the data, and you for your attention. Thank you.
Thank you. Questions?
I have a question for the first part.
Yes.
You were saying that data from bulk sequencing give you the golden standard for discovering SNPs, if I understand correctly?
No, so the SNPs we find using SNP arrays. So there's well-known population genetic there, and so you take the blood of those individuals and.
Yeah, I mean for detecting them in a.
Yeah, so.
Why is it supposed to be a golden standard? Why is it supposed to be better than what you discover from single cell?
Yeah.
Because I think single cell is supposed to be more sensitive.
Yeah, so it's better where the population is very homogeneous, like it is the case in IPS. So those cells are very, very similar to each other. Even when you see them, they cluster very, very closely. So when the population is very homogeneous, bulk is just less noisy. So there's just more power. On the other hand, when the more heterogeneous population, for example, the day 3 stage, if we had done even bulk using all the cells collected at day 3, that would not have worked very well, right? Because there's such heterogeneity, and then it doesn't work. And then in that case, single cells has more power because you can detect better, you can define better populations. And then, so it's, there's both these 2 arguments, and I think they're both right in different situations. So.
So the model you used was the, let's say, standard model, the same model that has been developed for bulk and has been applied to bulk, right?
Roughly, yeah. So it's a little bit more complicated than that because we had quite a few batch effects. So you'd have the same donor in multiple batches. And so in that case, if we were to average all cells from that donor, regardless of the fact they were coming from 2 different batches, that would have been a problem. So I'm actually taking a different mean per donor, batch, and day, even in this case. So it's a little bit more complicated than that. You can't just use it. And that's why it's very useful to have that random variable term with the population structure, because if you have, say you have 2 donors, cells from those 2 donors in 2 different experiments, and those 2 are different. You have 2 different average values, but it's actually the same donor. So in terms of genetics, it would be exactly the same. So you need to make sure that you tell the model these are, replicates. These are biological replicates of each other in terms of the genetics. So it's a little bit more complicated than that, but it's in practice an adaptation of the fast element.
And do you see any extensions that would be necessary or useful in terms of, I don't know, the noise pattern, the dropout? And
so there's many other things that we're trying. This is part of it. So I think mainly 2 things. In one sense, we're doing something that we call interaction Qtls. So it's something like the G by E in GWAS, for example, where you measure the effect of the combination of the SNP and, say, the lifestyle or some sort of environment the person is in. We do that for single cells. So the environment the cell is in could be the cell cycle state, for example. So we try to see, and that you can't do with bulk, right? So with single cells, you can really tell, okay, is the QTL only happen when cells are in G1 compared to S. that's one thing. And the other thing is, this is essentially always taking the mean. So it's something like bulk, so taking one average value. We are thinking of doing something like that with the variants instead. So we were trying to identify genetic variants, SNPs, that might affect the variability of expression from cell to cell instead of the mean. And again, I think that's only single cell provided, but it's part of, yeah. Oh, yes.
Very nice talk.
Thank you.
Could you maybe summarize once more, just for me, as I might be missing a part. So the utility of the single cell data, so you're basically using more or less, or you use approaches for your Qtl analysis which are modeled based on bulk-seq, but of course the single cell data gives you this resolution in terms of the dynamics of how the cell differentiates.
That's pretty much it. It's
anything else, but which is good.
Sort out
what's behind this.
I think yes, mostly is just sorting the cells in a proper way. So you can define what stage that cell is better than just trusting that every cell at day 3 will look like that, which is not the case. Yeah, so that's mostly. Yeah.
In this approach, you're using mostly population variations.
Yes, this is on germline mutations. Yes.
So I guess one could look at somatic mutations as well, and even induced mutations, like using mutagenic agents or radiations. Do you think that might be a little too harsh? Would this be also interesting?
Yeah, I don't know. I haven't really thought about it. So in order, for the stats to work, you need a lot of people with the same exact mutation, right? So this barely works with 130 donors. It will work a lot better with thousands of individuals. And then if you're putting in the somatic as well, there's even more variability and even less clear population. I think it might get a little bit tricky, but I'm not an expert.
You said with 130, it doesn't.
Yes.
Did you just say that, or how did you get at 130 now?
No, so this is how many donors we have in this. Yeah.
You said it doesn't work with less.
No.
I see.
Because we try. I mean, with 20, 40, 60, 80, we started getting something, very little, and now with more than 100, it's good enough.
I see.
Yeah.
But that's the number of donors.
Yes.
And you have many more cells.
Yes, but still the genetics, so in terms of sample size, you have to look how many identical, so how many unique genotypes you have.
But of course, for the somatic variation, it depends, right?
That will depend, yes.
It may work, let's say.
Okay,
more questions? Then thanks again.
Thank you.